Hello everyone. Today I will cover a very small topic on mycology. Hope you all enjoy the video and share and subscribe my channel. But most importantly, let's start learning about mycology. So, what is mycology? Fungus is a Latin word which means mushroom. In Greek, mushroom means myx. So, mycology, the term mycology came from myx and logos. Myx mean mushroom or fungus, and logos means study. So, what is medical mycology? We should know about we should know about the epidemiology, ecology, pathogenesis, disease course, diagnosis, treatment, before that investigations, prognosis of fungal diseases in human beings. And veterinary mycology, study of fungal diseases in animals. And what are the relations between these medical mycology and veterinary mycology? Zoonosis is possible. That means from animal to human transmission is possible. Example, sporotrichosis from Brazilian cats. Now, related terms and misnomers, mycosis means disease of warm-blooded animals caused by fungi. But mycobacteria, mycoplasma, and mycosis fungoides, those are only misnomers. Now, study of fungi includes molds, yeasts, mushrooms. Fungi are decomposers, pathogens, and symbionts. Fungi are decomposers, pathogens, and symbionts. They are more closely related to animals than plants. Now, why to study mycology? In the past, they were considered non-pathogenic or just lab contaminants. We have actually very little ideas about fungus and fungal diseases. Circumstantial immunocompromised background, they are emerging as opportunistic pathogens. As for example, dimorphic fungi re-emerged with AIDS, and then much more studies were held on these fungus. And agent of those are agent of biological warfare, industrial values in production of penicillin and enzymes like fungal diastase. An estimated 6 million species only, 6 million species are there, of which only 1,50,000 are known. Now, fungal classification and taxonomy. This is an ever-changing classification and taxonomy. There are Rosella species, Microsporidia, Neocalimastygomycota, Mycota, Chytridiomycota, Blastocladiomycota, Optidium, Olpidium brassici, and Zygomycota, Glomeromycota, Ascomycota, Basidiomycota. And of those, these Zygomycota, which has been changed now a lot, Glomeromycota, Ascomycota, Basidiomycota, those are important for studying medical mycology. Now, kingdom fungi, terrestrial organisms like molds, yeasts, mushrooms, which do not have chlorophyll, 10 phyla are there. Those are Chytridiomycota, Blastocladiomycota, Neocalimastygomycota, Zoopagomycota, Mucoromycota, those Glomeromycota, Ascomycota, Basidiomycota, Cryptomycota, and Microsporidia. So you can remember this with the mnemonic. Curious blasts navigate zoology mazes globally as Basidiomycetes creep mysteriously. You can remember those 10 phyla with this mnemonic, unless it will be very difficult to remember. Subkingdom Dicaria includes not last, but those two phyla, Ascomycota and Basidiomycota. Glomeromycota have two subphyla, Glomeromycotina and Paraglomeromycotina. All these are latest taxonomical classification to 2024 to 2025. Now, these special features, those are the lower fungi 1 to 6, that means Chytridiomycota to Glomeromycota. Those are produce those are asexual, those can carry out asexual reproduction, and they have flagella, early diverging, and they are simpler. And Ascomycota and Basidiomycota, those are higher fungi. They are terrestrial, and they reproduce by sexual reproduction also, and as well as asexual reproduction. and those have ascospores and basidiospores respectively. And they are dikaryotic, those two phyla. Complex and cryptic fungi are cryptomycota and microsporidia. Those are parasitic, asexual, or spore-based unusual traits are there. Now, what do you mean by dikarya? Dikarya, those are higher fungi. It's a monophyletic subkingdom or clade within the kingdom fungi, defined by a unique life cycle stage where two haploid nuclei coexist in each cell. That is the dikaryotic phase, N plus N. So dikarya includes two major phyla. Those points are to be remembered. And ascomycota, those are the sac fungi, produce sexual spores in sac-like asci. An example, aspergillus or aspergillus, you can pronounce like that, penicillium saccharomyces. And basidiomycota, those are club fungi, produce spores on club-shaped basidia. Example, agaricus, ustilago, etc. And what are the common features of dikarya? Dikaryotic stage produced N plus N phase before karyogamy. This is unique among fungi. Septate hyphae with well-developed cross walls are there. Those are the septa. And complex fruiting bodies like mushrooms, morels, truffles, etc. And genetic diversity, highly diverse group, over 90% of described fungi. Now, coming to the life cycle of ascomycota. Life cycle of ascomycota. Haploid ascospores are produced here. Haploid ascospores are produced in, and then they germinate, forming haploid mycelium in. Then occurs plasmogamy, fusion of cytoplasm that forms two different matting type. Fusion of cytoplasm of two different matting type. Matting type positive with red nuclei, and matting type negative has been shown with blue nuclei. And dikaryotic mycelium has been formed. It has N plus N nuclei, and then formation of ascocarp. These are the ascogonium, and this is the antheridium. And ascocarp is formed with dikaryotic hyphae. This is the fruiting body. And then development of ascus or sac-like structure. This is the ascus, sac-like structure with both blue and red nuclei. So they are dikaryotic and fuse to form the diploid 2N nuclei. Now, meiosis, which produces 4 nuclei again, and followed by mitosis with increasing number of nucleus up to total 8 haploid nuclei. Formation of 8 haploid ascospores inside each ascus. Now, ascus bursts, release of ascospores, and again the cycle repeats. And ascus is the defining features of ascomycota. Now, ascocarp types are perithecium, apothecium, and cleistothecium. Haploid dominant life cycle, both sexual and asexual reproduction possible. Asexual reproduction occurs via conidia. Now, from the spores from the conidia, and then asexual reproduction begins, formation of conidiophores, and those are the spatial hyphal branches. Conidia disperse, conidia conidiogenesis and production of conidia. Conidia disperse and conidia germinate on suitable environment, forming the new haploid mycelium. And the cycle again repeats. Conidia are non-motile asexual spores. Those are not formed in the ascus. Conidiophores are the spore-bearing. Conidiophores are the spore-bearing structures or the head end, and very common in molds like Aspergillus, Penicillium, and this cycle allows rapid clonal expansion in favorable environments. Now, the next is the life cycle of Basidiomycota. Now, asexual reproduction of Basidiomycota. In asexual reproduction, the haploid mycelium, again the same type. Haploid mycelium in asexual reproduction begins, and formation of conidiophores, production of conidia, which are the asexual spores. Conidia disperse, conidia germinate, and new haploid mycelium can be formed, and cycle repeats. Now, asexual reproduction is not common in many Basidiomycota, but found in rusts smarts, and some yeasts. Most Basidiomycota rely on sexual reproduction for long-term propagation. Now, what is the sexual reproduction of Basidiomycota? Uh, Haploid mycelium again formed, mating type plus and minus mycelia with each with single nucleus, and then plasmogamy, that is the fusion of the cytoplasm producing dikaryotic mycelium with n plus n nuclei. This is the plasmogamy occurs, and dikaryotic mycelium n plus n nucleus. Growth in basidiocarp, that is my example mushroom or fruiting body. Basidia develops on gills. Karyogamy in the basidium occurs. Basidia form diploid nuclei. Karyogamy occurs forming the zygote 2n. Formation of and then followed by meiosis 2n plus 4, and that is the 4 2n to 4 haploid nuclei, and formation of 4 basidiospores on each basidium. And now basidiospores released onto air, and germination occurs with new haploid mycelium formation, and the cycle repeats. Now the next one is the life cycle of Mucoromycota. Actually, this represents the life cycle of Zygomycota. This represents the life cycle of Zygomycota. Haploid mycelium mating type plus and minus, and specialized hyphae form the sporangia. Let's start with the asexual reproduction. Haploid mycelium, and first, this is the haploid mycelium. Specialized hyphae called sporangia develops mitotic production of haploid sporangiospores, and sporangium bursts, leading to dispersion of the sporangiospores, and new haploid mycelium again develops, and thus the cycle repeats. This is the asexual reproduction, very rapid and very common, allow fast colonization in moist conditions. Spores are non-motile, dispersed by air. And what about the sexual reproduction? Now, from the spores, mycelia develops two opposite mating type, positive and negative. They occurs plasmogamy. Each one is haploid, bearing one nucleus per cell, and then hyphal tips meet. Gametangia develops. This is the gametangia with haploid nuclei, and plasmogamy occurs, that is fusion of the cytoplasm forming the diploid cell, zygote. Meiosis inside the zygosporangium, the zygosporangium formation, and then meiosis inside the zygosporangium following karyogamy. First karyogamy occurs, and then forming the diploid nuclei, then meiosis, and zygosporangium germinate, and sporangium grows and produces haploid spores. Spores disperse and again germinate new haploid mycelium, and the cycle thus repeats, triggered by unfavorable conditions. Zygosporangium is resistant to drought and heat. Karyogamy and meiosis both occur inside the zygosporangium. Now, Glomeromycota, this has only asexual reproduction. This has only asexual reproduction. Spores germinate into germinates and then forms arbuscule. And example mycorrhiza, it grows around roots of plants. Now, this is the asexual life cycle of Glomeromycota. Large multinucleate spore, that is the glomerospore, germinate in soil, and hyphal growth occurs. Branched hyphae extend, and detection of root exudates. Directed growth towards plant roots. Attachment and formation of this arbuscule, that is the nutrient exchange. The arbuscule is for nutrient exchange, and fungus receives sugar, plant receives phosphorus and mineral. Optionally, formation of vesicles also will be there. New glomerospores form outside root in the soil, and the cycle repeats. This is the obligate symbiont, cannot complete life cycle without the host root. Arbuscule is the nutrient exchange hub. Entire cycle is asexual, no gamete, no karyogamy, and no meiosis. Glomerospores are thick-walled, multinucleate, and long-lived spore bearing spores. Now, oh, sorry. So, there are some points to tell about Zygomycota. Actually, the phylum Zygomycota is now considered obsolete in the modern fungal taxonomy, because molecular phylogenetic studies showed that Zygomycota was not a monophyletic group, meaning its members did not all share a single common ancestor. So it was split into several distinct genetically unrelated lineages. Now, Zygomycota has now been dismantled. We have seen in the classification of taxonomy and the classification on this slide. It has been divided into Zoopagomycota, Mucoromycota. Mucoromycota phylum are example Rhizopus, Mucor, and those have few subphyla like Mucoromycotina, Endogonals, etc. And Zoopagomycota has Zoopagomycotina. Extremely sorry, there was some technical problem. There is a misconception that today many fungal pathogens of previously Zygomycota phylum is now included in Glomeromycota, but this is not true. They have been reclassified into other phyla, mainly Mucoromycota and Zoopagomycota. So Zygomycota old classification, and now there are two phyla replaced those organisms, those fungus, which were previously included in Zygomycota. Why? Due to molecular phylogeny changes. Using DNA sequencing, and researchers discovered that Zygomycota was not monophyletic. Instead, there are two phyla. So, Rhizopus mucorlichthemia, those are Mucoromycota, includes many human and plant pathogens, and Basidiobulus, Conidiobolus, Entomopthora, those are Zoopagomycota, those are animal pathogens, parasites, Rhizopagus, etc. Those are Glomeromycota, those are obligate mutualists but not pathogen. So today I am ending my session. This is the part one, a small topic on mycology. I will meet you soon with part 2. Thank you.